lot of what I've been doing recently is looking at the distribution of carcinogenic minerals in different natural environments. And we'll go over three major types of carcinogenic minerals in this presentation. And then also a lot of, we're not going to go too much into exactly where the minerals are distributed, but it's actually a lot of challenges, and a lot of the pieces of this puzzle that I'm having to fit together come from like different boxes, or different people or different manufacturers are making the data to put everything together. So it's just becomes an interesting challenge in order to get all of this data to work together. But it should be a really interesting and helpful tool to have in the future for anywhere from homeowners to regulators to law firms. I mean, there's kind of countless applications for this particular type of research. Gene and I were talking earlier. He noticed that I also have PG at the end of my title. Back in October, I took the professional geologist exam, and I passed it. So as of December, I am a accredited professional geologist as well as a PhD. So I want to start talking or give a little bit of an introduction to some of the minerals that I want to talk about. Now, not all of these minerals are carcinogenic. Only specific varieties of these minerals are carcinogenic or cancer-causing agents. And all of these minerals are really interesting and important in their own rights. Each one of these minerals we have used for, or still do use, for different industrial applications. So a lot of these are economic minerals. They're still present in a lot of products and heavily used in a lot of different industries. So a lot of times when we think about some of these carcinogenic minerals, we think about our exposure to them in the built environment, so like in buildings and other types of products. But these are all naturally occurring minerals, so we're going to find them out in nature. But that's typically not what a lot of people, or that's usually not kind of like the first aspect that people tend to think about when they're concerned about the carcinogenic exposure of some of these minerals. So starting from the left over to the right, the first group of carcinogenic minerals I want to talk about are aragonite minerals. These are a subgroup of zeolites. Zeolites tend to be these kind of long, skinny minerals. They're used in water filtration, water purification, waste management, because they have like these large gaps down the center of the crystal that can accommodate for large cations like calcium or uranium. But those two images that I have, they look very different from one another. One's kind of like these long, skinny prismatic crystals, and the other variety is kind of these globular type of crystals. So there's several different varieties, but the specific variety of aragonite that is going to be carcinogenic are going to be the really fine hair-like crystals, and I'll show some pictures on another slide. And then quartz is the most common mineral species in Earth's crust. You probably have it on your hands right now. It's probably on your shoes. It's several places in your kitchen. It's probably in your mouth. Just because of there's just so much quartz that most dust in the air is going to be quartz. And that sounds maybe terrifying if you're thinking that I'm introducing this very common mineral in a carcinogenic talk. But I'll go over why some species of quartz are toxic and others aren't. And then the last two, serpentine and amphiboles. Serpentine is a sheet silicate, meaning that it grows into these plates, and you can split the plates off very much like you would peel sheets out of a book. And amphiboles grow into these really long, skinny prismatic crystals. However, both varieties can also form in fibrous habits. And kind of like with the aragonite, it's the fibrous habit that makes it carcinogenic, not the other types. So with aragonite, again, this is a zeolite mineral, and zeolites can be found in a number of different places. They typically form from the alteration of feldspar minerals, which are another common mineral in Earth's crust. But the aragonite ones specifically are found in altered volcanic soils. And erionite itself is not commercially used, but we use volcanic ash and volcanic rocks for numerous things, anywhere from like scouring agents to filtration to lightweight aggregates. And the carcinogenity or the toxicity of erionite was first discovered in a location in Italy where this one particular community was using some local volcanic ash in order to make bricks for their buildings and walls and walkways. And they didn't know that it was hosting this fibrous or this woolly variety of erionite, which was causing mesothelioma, which is a form of, it's an asbestos-related disease. So this is not, erionite itself is not asbestos, but it can cause some very similar types of symptoms that we find in people that have come down with asbestos-related diseases, a lot of times because of similar habit and similar chemistry. And so the images that I'm showing, these are used, or these were taken using a scanning electron microscope, so we can really view some minute, or we can get down to some really fine scales. I think the smallest scale that I see on here is about 4 microns. And to give people a sense of what that means is that 100 microns is about the width of a human hair. So some of these fibers are smaller than even a single micron. So some of these fibers are really small, or at least very skinny. They can be pretty long, but it's the skinniness of these fibers that makes them carcinogenic. And part of that is because of the surface area that is exposed. So the more surface area is exposed, the quicker reactions can occur inside the lung. And especially some of these minerals tend to be iron rich. And so iron will oxidize inside of the lungs, or it will react with the fluids inside the lungs to create basically this free radical generation. So you start mutating cells, which can lead to creating tumors and cancers. Now, in the image that I have showing on the left-hand column, those kind of fibrous woolly varieties, those are images of woolly erionite, but erionite also forms in prismatic varieties, like you see on the right-hand columns. So the woolly varieties have that high surface area exposure, and also the very fine diameter of the fibers can wiggle their way into different or deeper into the lung tissue than the prismatic varieties can. Now, inhaling a large quantity of any dust is not good for anybody. But it has been shown that the woolly varieties just work their way deeper into the lung tissues. So they do end up creating cancers and cell mutations, whereas the prismatic varieties don't. They kind of just sit around and weigh in the lung, but they don't create cancers or other types of, again, kind of mesotheliomas or potential diseases like that. Now, crystalline silica is another class of carcinogenic minerals, and this includes quartz and then 2 other minerals called tridymite and cristobalite. Now, quartz is, again, the most common mineral species in Earth's crust, and one of the databases that I'll be referencing later in this talk, I've included some of the data from that in the map that's showing, but the map that's showing is that all the dots or all of the gray dots across the United States are going to be soil pedons that were sampled by the USGS and tested for mineral content and chemical content. And all of the green dots in the map are locations that did not contain quartz. So all of the other gray dots across the map are soil samples that do contain quartz, whereas the ones in green don't contain quartz. So this is just kind of illustrating how widespread quartz is. Again, it's everywhere. And it's not just everywhere in the surface, but we use it for so many different things. And we use quartz-rich rocks or quartz-rich materials for countertops. Quartz sand is used in windows and fracking materials. Quartz-rich rocks are used in countertop. I mean, there's really a wide variety of different things that we end up using quartz for. Now, the carcinogenic varieties of quartz, one, they have to be really fine. So like a quartz sample or like a quartz crystal sample that might be sitting on your desk isn't carcinogenic because you're not going to inhale that. But any samples or any particles that are less than 10 microns can be inhaled into the lungs and into the deeper parts of the lungs where some of those cell mutations can happen. And as far as um, the toxicity of respirable quartz, not all respirable quartz causes cancer. We're actually not entirely sure why quartz causes cancer or is carcinogenic. It could be that it's a catalyst. It's a really hard, non-reactive mineral, so maybe, and it also fractures into these kind of curved surfaces, almost like broken glass. So it could act as a catalyst to kind of just tear up further linings for other reactions to occur. There could be some iron ions at the very surface of quartz, so freshly fractured quartz would then release those iron ions into the lung. But again, we're not entirely sure why quartz does as or causes as much damage as it does. So that's one kind of maybe goal further down the line of the study is to see if there are any trends in what types of quartz come from where that really lead into silicosis and other types of quartz causing diseases. Now, the mineral that I am most familiar, or the group of minerals I'm most familiar with, and where I'm really going to kind of focus this particular talk on, is asbestos. And asbestos is a commercial term. So this is kind of like the trade term that the industry uses in order to describe 6 different mineral species that occur with an asbestiform or fibrous habit. So asbestos, again, is more of an industry term as opposed to a mineral term. And there are 6 different minerals that can be classified as asbestos. So fibrous serpentine or chrysotile, fibrous riebeckite or chrysotolite. So chrysotolite is going to be the industry term. Grunerite and amosite. Amosite is also an industry term. And then there's asbestiform anthophyllite, tremolite, and actinolite. So the top one, serpentine or chrysotile, those are going to be sheet silicates. So you end up getting, again, kind of like these plates of minerals, but sometimes those plates can be really, really thin. So you're looking at a fiber. And then the other 5 minerals, those are amphiboles, and they tend to grow in these really long, skinny crystals. And under certain environments, they can grow to be super long and super skinny. So the image that I have, the kind of brown, tan colored crystal in the background is going to be a bundle of chrysotile fibers. You can kind of see how hairy they are, but they're pretty compact. And then the kind of hair, the hair one stranded across the front of that is chrysotolite. Chrysotolite also goes by the name of blue asbestos or Russian blue or some other industry terms. And amosite also goes by brown asbestos. So those are some of the common ones that have been and in some places are still being utilized in asbestos applications. So again, asbestos is a commercial term. It's the name of material that we end up putting into products, but it is a mineral, so it's naturally occurring. And typically these asbestiform minerals are going to form in metamorphic rocks or in areas where you have a lot of kind of directed hydrothermal activity moving into an area. And you can end up getting serpentine. Serpentine is going to form in metamorphic rocks, so in areas where you have really intense heats and pressures. And then amphiboles can actually form in igneous rocks, so they can crystallize out of magma or out of lava. And they can also form in metamorphic rocks, and they can also persist as sediments. Out of the bunch of these, amphiboles have the slowest reactivity, so they tend to last longer in the lungs, which makes them the more hazardous varieties of asbestos because they just take so long to break down and start reacting. And a lot of times, cases of mesothelioma or other types of asbestos-related diseases that are spurred by the amphibole varieties of asbestos, they're not noticed until maybe decades after the person either like worked at an amphibole or like an asbestos manufacturing plant or mine, because it just takes so long for the effects of those minerals in order to be seen. Whereas out of the bunch of those, serpentine is going to have the lowest or the quickest reaction time. So you'll start to see those symptoms quicker, but the asbestos ends up degrading faster inside of the lungs. So those symptoms don't persist for as long as if it was an amphibole species. So a lot of asbestos studies that have been done, they don't differentiate between the chrysotile and the amphiboles, or at least when we're talking about asbestos particles in the soils. These are different minerals, they grow in different environments, and they have, again, they have very different reaction times and behaviors inside of the body. So it's important that we split them up into two separate categories. I know these are a lot of words, but I want to walk you through some of the databases that I'm using. Now, these are all national databases, so have been compiled for across the United States. They're also open access, so anybody can go to these links and access each of these, or the information in each of these databases. And some of them are, well, they're all quite extensive, some more than others. So one of them was the National Cooperative Soil Survey through the USDA. And this one's pretty extensive. An individual layer of soil inside of, kind of like a cross section would be called a horizon. And each of those cross sections, or kind of like bulk of soil, is going to be called a pedon. And so there was a number of these soil samples that were collected and still are being collected through the NCSS. And the mineral content of each horizon is determined mostly by a point count. So you end up looking at a spread of sand grains underneath a light microscope, and then some poor lab tech ended up having to count out 300 grains and identify each of those as what type of minerals. And so the mineral content for each soil horizon is identified in percents based off of those 300 grains. And you're able with light microscopy, you're able to identify individual species of amphiboles. So for instance, again, 5 amphibole species are classified as asbestos, but there's almost 100 species of amphiboles. So not all amphiboles would be regulated or are considered to be asbestos, even though all amphiboles have the potential to grow in that fibrous habit. So some of those individual amphibole species have been determined or differentiated inside of this database, but a lot of these minerals, you don't know the precise species without doing a precise chemical analysis of each of these minerals. And something similar with the serpentine minerals, there's actually 3 minerals in the serpentine group, but only one of them is fibrous, and that's chrysotile. And in some of these cases, you can differentiate the different serpentine group minerals from each other, but in some of these analyses, you can't. And so that was another kind of struggle, is some of these databases don't differentiate the different species of amphiboles, and they also don't necessarily differentiate the different morphologies. All right, and so then I had mentioned earlier that I kind of keep coming back into this particular project, and sorry, I just knew what picture was coming up. So the one on the left is me back in grad school, and my very first trip as a graduate student was traveling to the very exotic location of Lincoln, Nebraska, because that is where the USDA houses all of their samples from the soil survey. And so they just have, gosh, I can't even remember how many warehouses they have of these samples. And you can't even see all the way in the back of this image because of just how extensive this particular basement is. But so all of those boxes are gonna represent a layer of soil. And so they're all coded. They all have the sample number, the pedon number, project numbers, all that other information. And so so it's kind of like this library or this catalog of soils from across the United States and across the world. So you can essentially check out these soil samples, as well as some prepared epoxy mounted slides like you see over in the right-hand image. And so I like this particular soil sample slide image simply because it just demonstrates the potential kind of variety of minerals that you can find in a single soil sample. One of the other studies that I was looking at is a uh, geochemical and mineralogical database of soils for the counterminous United States, done through the USGS, or the United States Geological Survey. Now, this one, in the soil survey, it's basically the samples are collected wherever soil scientists can get them or wherever there's places of interest. Whereas in this particular database, they wanted to see the distribution of soils and how they change across the United States. So they took a lot of time and attention into selecting sites before they went out and sampled. So for instance, they wanted to do a site per every 1,600 square kilometers. And they also collected samples from just the top 5 centimeters, the A horizon, which is the most altered horizon of a soil pedon, and also from the C horizon, which is going to be the least altered horizon within a soil pedon. And from here, the mineral content was actually determined using X-ray diffraction, which is one of my favorite techniques. It's using X-rays and kind of, you're not reflecting them, but you're moving them through a crystal to see what angle those X-rays get refracted or diffracted back through the crystal out to a detector. And that angle will tell you about the spacing in between atoms, which is characteristic of different groups of minerals. Now, again, it's characteristic of different groups. So you can use this type of technique in order to refine all the serpentine group minerals. And you can also use this technique to identify certain subgroups of amphiboles, like hornblende. So hornblende is a subgroup of amphiboles that all have very similar chemistries, but there's something like 20 species of amphibole inside of the hornblende subgroup, and only 2 of those are regulated as asbestos. So you can't exclude hornblende from these types of studies, but at the same time, it's so broad, it's going to include a lot of other species aside from just the 2 that are regulated as asbestos. So this is, again, a similar map as what we were looking at when we were talking about quartz localities. But so all the dots here are going to be all of the soils that were sampled across the United States for this particular study. So again, really ubiquitous, or really even distribution, or at least as even as they could possibly get it throughout here. And then they also have, you can access all of this data through the MRData database at USGS, and you can look at heat maps for all the different minerals. So for instance, this is a screenshot of the location or the existence of hornblende inside of the A horizons of soils. So the redder colors are going to indicate kind of hotspots or higher concentrations of those amphiboles, or this particular subgroup of amphibole. Now, luckily for us, especially in regards to asbestos, some of this work has already been done as far as weeding out where the occurrences of asbestos minerals themselves are. So Brad Van Goosen from the USGS put together a series of different publications and kind of tied them all up together into a 2019 publication. And it's a list of all the occurrences of asbestos throughout geologic literature, throughout, again, all across the United States, and even lists whether the occurrences are large commercial mines, or if they're small, maybe veinlets or small prospects, or even if they're just locations of known existence, not necessarily something that was mined for. But the neat thing about this database is he does specify the asbestos type as best as he can, along with the type of rock that the mineral is hosted in, which is really key because with another database that we ended up using, we don't quite get all this information. I'll talk about that in here in a second. So this is a screen capture of kind of like the online portal that you can access of this database through USGS. And that's nice. You can access all these databases through the USGS, but you can only do them one at a time. There's really not a way to overlap all these databases on top of one another unless you download the databases separately, like what I've done, but you can't view them all online. And so here we're looking at all the locations of the red squares are mines, and the green squares are prospects. So these are going to be kind of like smaller, kind of smaller scale operations. And then the white squares are going to be, again, just maybe some other noted occurrences of asbestos, but have never been economically exploited. So the USGS also has this mineral resource data system. This is a worldwide mineral resource database, but you can chop that down to just look at the United States. The problem with this is a really extensive database, but the problem is it was actually a melding or a mesh of two separate databases that were combined during the 60s. And they're being updated, but because of all of the economic mineral resources focused out in the western part of the United States, they're starting more over there and then working their way over to the east. And also, all of these mine sites are basically just categorized as what type of commodity is being exploited. So again, we're coming back to more industry terms as asbestos. They're not designating what type of asbestos, if it's chrysotile versus amphibole. And so this would be a screenshot of that particular mine. So again, I'm going to take all this information and overlap it into a few maps. Now the last one, I know I don't have a whole lot of information on this one, but the last one is a state geological map that was combined again by the USGS. And you can end up searching the database for all the different rock types. And my goal here is to help use this database to see what rock type some of these mines are hosted in, and see if we can find a correlation between what types of rocks and what environments and some other information to see if we can tie everything in together to determine where there might be some other locations, some other rocks, or even other soils that might be hosting asbestiform minerals. And so this is a very generalized schematic of the map. And as you can imagine, this is a state geologic map compilation. So all of the state geologic maps that were done by each state's individual geological survey have basically given or donated their maps or their data over to the USGS to be able to compile into this one gigantic map. And so the reason why some of these states kind of stand out a little bit more than others is that some states are going to put more or they're going to dedicate more resources and time into making detailed maps as opposed to others. So that's why you can start to see some outlines of individual states in here. And again, that's just going to be because of the resources that each state can put towards the makings of these types of maps. Now, with some of the preliminary data that I have, I've started to see some really interesting trends. But so this particular map, the orange circles, we're looking at deposits of known amphibole asbestos from the Van Goosen 2019 database. So all the orange circles are going to be places where we know amphibole asbestos exists. And then all of the kind of yellow squares, those are going to be soil horizons, the topmost soil horizon that contains greater than 1% amphibole. And that's a compilation from the Smith 2013 database from the USGS and the USDA's National Cooperative Soil Survey. So think of the orange circles as being definite locations of amphibole asbestos, and then the squares as being potential locations of amphibole asbestos to be located inside the soils. So there are some definite trends of where we're finding amphibole asbestos, but amphiboles in the soil is pretty ubiquitous across the United States. So we can't just simply stop at regulating maybe soils to have or to contain asbestos and just simply calling them contaminated if they contain amphiboles. We need to kind of go a step further because again, otherwise we end up regulating a large part of or most of the country. So then stepping out and looking at chrysotile or serpentine. So again, the dark green circles are going to be all the locations where we have known deposits of fibrous serpentine or chrysotile. And then all the green squares are going to be all the locations where we have some type of serpentine group mineral inside of the soil. Now, there are more prospects or more mines of chrysotile asbestos than amphibole asbestos. And part of the reason for that is just the chrysotile bundles tend to form in bigger bundles and longer strands. So when we're using asbestos to make fireproof textiles, then we have more material in order to weave into a textile. Whereas the amphibole asbestos, they tend to make smaller or shorter fibers. So typically we've used those for spray-on insulations, floor insulation, and pipe insulations. So there is a difference in kind of like how many mines or prospects we have of each mineral because of their economic importance. Now, the reason why that there's so few locations of chrysotile in the soils, again, is because chrysotile breaks down pretty quickly in not just inside of the lungs, but in reaction to water and humid environments. And serpentine is it does have some hydrogen and oxygen inside of the structure, so you're not going to find it in really hot environments, but it will break down under lighter temperatures and pressures. So this particular mineral forms at higher temperatures and pressures than it's used to here at the surface, so it starts to break down. Whereas amphiboles can form in cooler conditions, so they're happier up at the surface, so they'll stick around longer before they start to break down. If we put all of this data together, so all of the black circles are going to be deposits of known chrysotile and amphibole asbestos. So those are all of the locations in Van Goosen's 2019 publication database. And then all the white squares are going to be all the top soil horizons that contain greater than serpentine and amphibole minerals. Now, again, there's a lot of challenges here, but we definitely start seeing trends between where there are asbestiform minerals and then where there are soils that are hosting these different types of minerals. So we can't just stop at just looking at soils that contain amphiboles and serpentine, because that's going to be a lot, and we can't regulate, again, that much large quantity of land. But this does give us, I guess, a clue or a hint as to what kind of soils we can start looking for. And the reason why this becomes important is because there have been cases where developments have begun in areas not originally known to host asbestos, and then they were found later to have hosted. So, for instance, there was a community in California. They broke ground in one particular area in order to build up a residential community, and had already sprung up houses, already sold houses. People had started moving in when they found out that there was actually some chrysotile asbestos inside of the soil at that particular site. And so then they had to figure out a way to remediate and clean up a lot of the soil in people's yards, again, in order to get rid of that particular hazard. So again, some of the challenges that I've run into in assembling all of these databases and where we're going to move forward from here is that not all of these databases are going to differentiate between the different varieties of these minerals. So that's going to have to be a piece that we figure out is, are there some other context clues to tell us what type of mineral are we probably looking at? What type of amphibole is present here, or what type of asbestos? Again, to kind of put all those context clues together, and that'll also help us figure out kind of like what soils to look at even more. So the example I have is that tremolite and actinolite are two of the regulated asbestos species that are in the broad hornblende amphibole subgroup. So again, there's, I think, something like 20 species of amphiboles inside of the hornblende subgroup, but only 2 of those are going to be actually regulated as asbestos, even though all of them have the potential to form in that asbestiform habit. And not all elongate particles are fibers. So a lot of times when they start regulating for these asbestos particles, the easiest way to tell somebody to count how many asbestos particles you have in a sample is to count particles that are 3 times or 5 times longer than they are wide. So you're just looking at aspect ratios. And this could be helpful in some contexts, but if you're just telling somebody to go off and count the number of items that are 5 times longer than they are wide and count them as a fiber or as asbestos, if you're sitting in my kid's art space, for instance, every single marker that I'm looking at on that art table would then be counted as a fiber. But no one's concerned about inhaling those, again, down into your lungs. That'll cause other problems, but it's not going to get inhaled down into your lungs. So not all elongate particles are fibers, and some of the earlier microscope images that we were looking at, there is a difference in shape and surface area between the fibers and the long skinny prismatic crystals. And also, not all fibrous amphiboles are regulated as asbestos. So not all, again, all amphiboles have the potential to be in a fibrous habit, but they're only regulated by industry, by OSHA, and by other groups as being carcinogenic if they are one of those 5 species, not all of the almost 100 species of them. So every component of this study has been absolutely complex, not limited to some of the definitions used for these minerals and morphologies. I'm sure that every one of you, when I say the word fiber, has a very specific image in your mind, but I guarantee you that the way that you define fiber is going to be different from everybody else that's sitting in on this talk. And that's part of the problem, is that not just different people have different definitions for the word fiber, but mineralogists tend to define a fiber differently than people in the textile industry versus the people in like an insulation kind of industry. So different industries have different definitions for each of these particular types of particles. And even the formation environments of these minerals are complex. So serpentine, out of the group of these, serpentine environments are pretty straightforward. You need a magnesium-rich rock that undergoes metamorphism with moderate temperatures, high pressures, and a lot of water. And so there's only a few different types of places that we can, or few types of settings that we can find those different types of rocks. However, amphiboles grow and form and persist in just about every rock type. And we're finding that we don't just need high pressures in order to make the asbestiform varieties. There's a lot of other factors that come into play. The national databases used in this study are entirely complex, and trying to determine what information is pertinent to this particular study has been quite a bit of a challenge, because we're finding trends in several aspects that we didn't think to look through before. So for instance, there are some trends between chromium levels in the soil related to the serpentine levels in the soil. So some of the context clues that we might be looking for aren't even mineral species. We might end up looking for different concentrations of other elements. And the last challenge, and probably the absolute biggest and the one I always struggle with, is just the relationships between every faction that would benefit from this particular type of study and toolkit, is that there's already kind of a strained relationship between industry, regulation, and the general public. And to be able to educate all 3 of these different groups into how to use a database like this, or any of these databases on their own, has constantly been a struggle. So hopefully, if we can bring in more data and put it all together into this kind of visual context, we can better describe the problem and kind of better determine where some of these locations of natural occurrences of asbestos are, and other carcinogenic minerals. So here at the end of my talk, I do want to give a very big special thanks out to Graham Walker, who is one of my undergraduate research students last summer. He helped me compile a lot of this information. He did a lot of kind of like the initial database imports and setup, which it was monotonous and tedious, and I'm glad that he did it because he did a great job. And then also to Dr. Thompson and Dr. Langle, both of them have been instrumental in both support in the GIS programs that I've been using, as well as emotional support for any type of GIS program that anyone uses. So again, I'd like to give a big shout out to all three of them because they've been instrumental in this particular project. And with that, I would like to say, big thank you for all of you coming out. Haha, no pun intended. But I would love to take any questions or comments from anybody. So thank you.
Brittany, I have a question.
Sure.
So, all right. So as I wrote it down because I wanted to make sure I expressed it correctly.
Okay.
I'm not a geologist, so I find all this like super fascinating. So as a biologist, let me turn my video on. I turned it off so that I wouldn't distract from your program. Okay, here I am.
Hi.
All right. So as a biologist, I'm accustomed to thinking about properties of living organisms as adaptations. So
Yeah.
if I saw fibers in a plant, I would think that they served some useful function to the plant.
Sure.
So I find, all the photos you showed of all these fiber structures and these minerals.
Yes.
I think that's so interesting, but all I keep thinking of is, why do minerals have fibers? And maybe you can't think about minerals the same way you think about plants. So do the fiber structures serve some purpose?
Well, so
or are they just there?
But that's an interesting, that's a very interesting question. And there's a whole, not brand new, but there's a relatively new field of like evolutionary mineralogy that's come up to kind of help answer some of those questions. But for the most part, it's not so much that these minerals are adapting to serve a function. They're responding to the environment that they're in. So these atoms simply just want to assemble in a very certain way. So then if there are these strains or stresses that are forcing them to go in one direction, then like with the amphibole crystals, instead of making these big prismatic crystals, they're going to make these very, very long, very skinny crystals instead, because those atoms are forced to stack in a certain direction.
So it's really a response to environmental stresses, like maybe physical stress on them.
Yes.
Oh, okay. Well, that's interesting. Thank you.
No problem. Any others?
Brittany, I guess I do have one more question.
Sure.
Of all the minerals, I know that you study asbestos a lot, but of all the minerals you talked about, which one do you think is really the most like interesting? I mean, I know you study asbestos, but sometimes we study things just because we have an opportunity to do so or we got funded to do so. But are there any that you think are like, the most curious, or like if you could study it, if you had funding, that you would go for it?
That's an excellent question. And out of all of those, I think I find the toxicity of quartz to be the most fascinating, simply because there's so much we don't know about it. And again, it's one of the most common minerals. So everybody is inhaling quantities of quartz, respirable quartz on a daily basis. So why aren't there more cases of silicosis? Is this simply a dose question? Like we just need to be inhaling absurd amount of silica, or is there something else specific to the silica that's being respired? So that would be interesting. I would also have to find probably an epidemiologist friend or colleague.
Yeah.
No, but that's an excellent question.
I had no idea I was inhaling so much quartz. I never thought about it.
They also use quartz, fun fact, as like a declumpifying agent in a lot of spice blends.
Really?
Yes.
Wait, does that mean we should be like grinding our own spices?
Yes. No, they don't put a whole lot in it. It's just to keep some of them from clumping. And so like some of the spices, if you notice, like they just feel extra crunchy for what's in there, is because there's some silica sand in there. So if anything, it's just wearing down on your teeth more. Well, I appreciate all of you coming out, and I'm sorry you can't see me.
There's a question in the chat. Can you see it?
Oh, do you have any regulations that we should be looking towards creating in those communities with large concentrations? That's an excellent question. And I think some of the big challenges with that are, before we do any developments, we should be doing mineralogical studies on the soil. Like we do that anyways, for different reasons. One, you don't want to be building on a soil with a lot of shrink and swell clays, or you don't want to be developing on a soil that's really high in concentrations of certain types of elements like uranium. But as I would say that what we need to add to the regulations that we already have in place are to maybe open that up to other types or species of amphiboles or other types of materials. Because again, a lot of times when they're looking at those soil samples, they're counting fibrous materials, and they're just counting how many fibers they find. And Irene, as you mentioned, there's a lot of biological materials that form fibers or fibrils. And a human hair found in a soil sample would be counted as a fiber, but that's not carcinogenic, or at least I hope it's not. Otherwise, we're all in trouble. So I think as far as regulations go, there just needs to be more research on what materials really are toxic, what makes them toxic, and how can we identify them.
Well, thank you, Brittany.
Thanks, Irene.
Enjoyed it.
Yeah.
I'm glad you all could come out. Have fun.
It's great hearing your voice.
Right. Thanks, Irene.
No, thanks a lot. Enjoyed it.
Thanks everyone for coming.
Thanks, Jane. Thanks, Jen.